Thank you very much for this wonderful opportunity to present at this webinar of yours. And as Bettina mentioned, I'm going to give you a little bit of insight on our latest microclot and platelet pathology research in long COVID and how we can gather our strengths, manage our challenges, and also elevate our excellence with different collaborators. So the research and collaboration discovery science are always done with a dedicated team of researchers and collaborators and postgraduate students. Too many to name, but just a few to point out here. Some of the collaborators from this presentation is Professor Douglas Kell. He's my long-standing collaborator from the University of Liverpool. Then my Stellenbosch team, Dr. Chantal Venter, who was the blood lab manager, and just a few of my dedicated students. I'm also very happy to report that this wonderful team of Bettina have been added as a collaborator, and hopefully soon her name will also be on this list. Today I will share with you the following discussion points. The latest hypotheses on the pathologies involved in long COVID, and also if we look at any CFS. The debate if microclots are indeed clots. Latest research of other groups, the presence of microclots in other inflammatory diseases, and the question, should we care that these deposits are present or not in blood, in circulation, and then some novel methods to study amyloid microclots. However, just to give you a general and quick recap, focusing on the scope of our research over the years, we are interested in the pathological blood clotting involving the main clotting protein fibrinogen, which is a soluble protein in circulation when you are healthy. We have shown that soluble fibrinogen molecules may become amyloid in nature when they interact with inflammatory molecules like the spike protein and others, such as LPS, etc. Fibrinogen then takes on a fibrinoid structure, and we have shown this with various amyloid protein markers, including theophlavin T and also amitracker. We call these anonymous amyloid deposits microclots, and we only started calling them microclots in 2022. One of our first experiments where we showed the amyloid nature of fibrinogen was in 2016. Here you can see in a controlled platelet-poor plasma sample exposed to theophlavin T and thrombin. Thrombin was added to create an extensive fibrin clot. As mentioned, theophlavin T is our marker for amyloid protein, and none of this very bright spots of THT can be seen in a healthy clot. However, in B, where plasma was pre-incubated with LPS, followed by exposure to thrombin to create the clot, you could see significant microclot formation or clot changes, structural changes. And in C and D, plasma was pre-incubated with LPS again, but we also added lipid binding protein. This lipid binding protein binds LPS and it prevents its interactions on fibrinogen. Thus, very little amyloid changes can be seen in the plasma. THT was added to this as well, and now the signal was not there anymore. In 2019, we studied the acute phase inflammatory molecule serum amyloid A. We added it both to plasma from healthy donors and also to purified fibrinogen. Here is an example of correlative light electron microscopy, also called CLEM, to detect the serum amyloid A. What we did is we added serum amyloid A antibody, and that is the green signal, and we added our amyloid molecule dye to find amyloid, which is amitracker, and that is the red color. So amitracker is very much the same as THT or theophlavin T. It also detects amyloid protein, and this is in purified fibrinogen. So if you look at the areas here, you could see the red signal, the amitracker, and you could see the green signal, the serum amyloid A antibody. This suggested that the serum amyloid A interacts with the fibrinogen and causes it to form amyloid areas within the purified fibrinogen areas. We also added serum amyloid A to a fluorescent or FITC tagged purified fibrinogen. This you could buy from Sigma. This fibrinogen is green, and we added thrombin again to create an extensive fibrin clot. In A, on the top left, you can see the clot that forms in the presence of this FTSE fibrinogen with thrombin, and it's a very light green tinge, and you would see the fine net. This is what you would expect in a fibrin clot. In B, serum amyloid A was added, and it formed a matted area in some dense areas of the clot, then resulted. In C and D, we also added our AMI tracker, which is amyloid marker, and you can see that the amyloid area coincides with that of the dense areas of serum amyloid A. So that is the reddish signal. On the right, you can see micrographs where we added serum amyloid A to whole blood of healthy donors. So A to D is without serum amyloid A. So this is how the red blood cells and platelets would look like. And then you could see the amyloid areas that form if you look at the latter figures, where you could see that serum amyloid A actually causes significant platelet hyperactivation. If you look at the platelets in J, for example, and you could see it even looks as if the red blood cells are glued together by the fibrinogen that now have been changed in the presence of serum amyloid A. So this is just some of the examples of many of the papers that we wrote to discuss changes in fibrinogen with added inflammatory molecules. So in short, we studied platelets and red blood cells and fibrinogen and how they interact with inflammatory molecules that may also be related to endothelial damage. So fast forward to the pandemic, we turned our attention to acute COVID-19. During the early stages of the pandemic, we could not have access to thrombin from our blood bank. Thus, I decided to only add theophlavin T, our amyloid marker, to platelet-poor plasma. And here you can see the differences in platelet-poor plasma protein structures in acute COVID patients, where we compare the plasma smears to the acute COVID patient to that of healthy and also patients with diabetes. Here you can see the microclot formation demarcated again as the green signal from theophlavin T, which is our marker for misfolded protein. We found small areas of theophlavin T signal in plasma of healthy individuals. And remember, this time we didn't add thrombin. So it is simply the plasma with added theophlavin T and under the microscope. And you could see this in the micrographs on the left. More abnormal microclots were noted in plasma of individuals with diabetes, as one would expect. What was, however, quite significant to us was the extent of the abnormal microclotting in acute COVID plasma, as seen on the far right plate. Also note the scale bar at the bottom of the micrographs. We also looked at blood clotting in acute COVID-19 using our scanning electron microscope. We found that platelets were damaged and clumped together. It seems as if they were broken open. And the red blood cells clumped together. Clotting protein that is supposed to be soluble formed these spontaneous, dense, matted areas, microclots. In the micrographs on the far left, you can see platelets that are really hyperactivated. Note the yellow arrows. In the middle are micrographs where platelets are attached to red blood cells. Note the white arrows. And on the far right, you will see micrographs of spontaneously formed microclots that, as I mentioned before, glue together the red blood cells. Note the blue arrows. So let me return to the discussion point and the first one. The latest hypotheses on the pathologies involved in long COVID and also ME/CFS, perhaps, as this is also a post-viral disease. In a nutshell, there are various hypotheses on the causes of long COVID, and the role of abnormal clotting and vascular damage also plays an important role here. In this overview figure, we show the key pathologies, which are the black arrows involved in long COVID, as well as their interactions with each other, and those are the red arrows. You can see in A, thrombotic endothelitis and anomalous amyloid deposits or microclots play a significant role in our mind. Immune dysfunction and autoantibodies, viral persistence, cytokine dysregulation, muscle involvement, hormone dysregulation are all part of the phenomenon of long COVID. And then there might even be subtypes of long COVID as a result of the key pathologies driving the symptoms. The next discussion point is the debate. The debate is are these microclots indeed true clots? You might be aware that this is a debate among some, that the dense amyloid deposits we refer to as microclots, are they indeed clots? The question, therefore, is, are these dense matter deposits simply something like an amyloid particle that are merely present in plasma, but it doesn't have any meaning or reason to worry about? It's just there. We must just ignore it. Or that perhaps if we need to answer this, we could ask the question, is this a philosophical question that we need to debate? Are these microclots present? And why are they there? Is this perhaps a better question to ask? If they are present in inflammatory diseases in higher concentrations and size ranges than in controls, is this a reason to ignore them? So let us first look at what is recognized by the experts as the definition of a true clot. A true clot is mostly seen as a structure that is involved causing a severe event like a heart attack or a stroke. The composition and structure of such macroclots can vary depending on its location in the circulatory system and the underlying conditions that led to its formation in the first place. An important component in the plasma protein fibrin and fibrinogen that entraps various red blood cells and the white blood cells and the platelets. So that's a definition, the general accepted definition of a clot, or then a macroclot causing a thrombotic event. So returning why we say our microclots are indeed microclots. In a proteomics analysis in 2021, we digested the microclots that we used to study, and we used a double trypsin digestion method, as the microclots turned out to be quite resistant to enzymatic digestion. During the data analysis, we discovered, amongst others, various molecules entrapped inside these microclots, including alpha-2 antiplasmin, von Willebrand factor, complement proteins, and the various fibrinogen chains, and also coagulation factors that were trapped inside the solubilized, after the trypsin digested, fibrinolytic resistant deposits or microclots. Here is the clotting cascade. It was just interesting for us to note that alpha-2 antiplasmin is a molecule that is known to prevent clot breakdown. And that might be the reason then why we saw that the microclots were fibrinolytic resistant. A next qualifier of a clot could be that the red blood cells become entrapped inside a fibrin mesh. Now, if we turn back to what we have shown and looked at in whole blood of patients with long COVID, here we can see microclots, or these deposits, in and on red blood cells. So one could come to the conclusion that it is the start of an entrapment of the red blood cells, and thus a start of a clot. We could then say it might be a microclot, albeit not a large thrombus yet, but perhaps on the pathology pathway. Here is an example of a red blood cell of a healthy individual versus a few glued together microclots in an individual with long COVID. So one could suggest that red blood cells are entrapped and entangled in microclots. Again, a larger magnification of a red blood cell showing significant microclot deposits on the red blood cell membrane. This will most definitely impair erythrocyte function, flow capacity, and their ability to transport and deliver oxygen, and also remove CO2 in the capillary beds. Here is an example of a microclot in whole blood of an individual with long COVID, once again in whole blood. So back to the discussion points. Let us look at the latest research of other groups and also the presence of microclots in other inflammatory diseases. Since 2021, lots of new discoveries followed, not only in our lab, but also in the labs of others. In a recent Blood Advances paper from the TOE lab in the UK, the team used amyloid marker theoflavin T and proteomics to look at microclot composition in the disease sepsis. You could just imagine that we were very happy to read in their conclusion, and I quote, "Overall, we believe that this is an important step forward in the potential utility of this assay, the microclot assay, as a clinical, useful, and practically measurable biomarker to identify sepsis, predict DIC and mortality in the ICU." Here is a fantastic paper from the Wust group that many of you might know. In their 2023 Nature Communications paper, they found amyloid deposits in muscle using our methodologies of theoflavin T detection. If we agree that ME/CFS is also a post-viral disease, perhaps there are lessons to be learned from acute and long COVID for this disease as well. And I believe that the Wust group is looking at ME/CFS as well. The focus of our research group, therefore, has been also to see if we could help other researchers and labs to be able to use the microclot method. And if we just look at this results from the wonderful Dalton group, they focused their group the past months to look at the methods to validate the microclot method with an automated system at the University of Sheffield. And then the Yonker group from Harvard recently found evidence of microclots and activated platelets in pediatric long COVID patients. Interesting microclot formation is not only found in acute and long COVID, as I mentioned, but also in many other inflammatory diseases where clotting pathologies are present. And this just circles back to what we have found previously in our research, where we found these microclots on red blood cells in rheumatoid arthritis, lupus samples, and even Alzheimer's type disease. So now the question that arises, should we care that these deposits are present or not? And should we indeed call them microclots? I think I will leave that question for you to answer. I think I have given you some information, and I think the debate is still open. Should it be called microclots or not? So let's now look at novel methods to study these amyloid microclots. In 2022 and early 2023, we crowdfunded to obtain an imaging flow cytometer, and we were very happy to have, we received this imaging flow cytometer in our lab, and we received it after a very short time of crowdfunding. And I must really thank patients and clinicians and researchers who contributed to this crowdfunding initiative, and Polybio Research Foundation, as well as Kernels, as well as Barbie Research Foundation. So we are happy to report that in 2023, our first long COVID and control microclot imaging flow cytometry method was published. And we hope that this would be useful to quantify microclots in plasma samples, other than using our first method of detection, the microscope. Just some of the mean data, area data, and objects per mole data to show that there are significant differences between controls and long COVID samples. This was from our first paper. But here are also some results where we compared long COVID clot load to controls and also ME/CFS. We found that the number of microclots per milliliter is still most in long COVID samples, but that ME/CFS is still higher than in that of healthy individuals. And that is what we would have expected. If you look at this slide, here are the distribution of log transformed object counts within specific area ranges from flow clotometry or flow cytometry analysis across healthy participants, our ME/CFS, and our long COVID groups. The density plots overlay the scatter plots, and they show the distribution of data points with medians indicated by the line within the boxes. If you look at the panels, panel A is the count 0 to 100, B is the count 100 to 400, C 400 to 900, and then above 900 to 1,600 and plus. The counts underwent natural logarithm transformation to accentuate underlying patterns within the area, the data, and to enable interpretable visual displays. And this is the log count medians that are displayed. Recently, we also started a collaboration with Professor Allard Thierry, and he's from INSERM in France. He has a keen interest in net formation, so neutrophil net formation. And he also worked with cancer samples, and that's where his interest in nets and neutrophils came about. So he's really a world expert in this phenomenon, and he approached me during 2023, and we had a collaboration agreement where I sent samples to him. So just a little bit of a background on nets. Nets are composed of chromatin and of several proteins derived from granules, and these might be neutrophil elastase, myeloperoxidase, bacterial peptides, cathepsin G, lactoferrin, matrix metalloprotease 9, etc., each contributing to the elimination of bacteria. This allows nets to eliminate microbes within the first hour of infection. This is the general hypothesis of and known experiments of nets. So doing if we look at these nets, they also have DNA fibers and all chemically interact in the nets. And DNA is the main net component, and the principal net byproducts consist specifically of degraded and fragmented DNA that are shed into the bloodstream in the form of circulating DNA. So we indeed found that nets are entrapped and structurally associated with microclots. And this paper is currently under review. So what we did is we took blood samples, platelet-poor plasma samples from long COVID samples, and we exposed it to various markers for nets, which is the Hooks, the blue signal, and also Cyto here. In this case, on the screen, you can see it's a red signal that stands for DNA. We used our theoflavin T marker again, which is the green signal for amyloid microclots. And then you could also see a light blue area for MPO, myeloperoxidase. So our conclusion was that indeed, inside the microclots, the circulating microclots, nets are entrapped, and they might overlay with the amyloid protein areas, but they also might appear structurally separated. And this is a wonderful machine learning micrograph that Alan and his team produced for us, showing the interactions of the various proteins with the amyloid areas of the clot. So as I mentioned, the paper is currently under review. We also had experimentation on the long COVID samples where we looked at the content of the various molecules in his lab. So as I mentioned, samples were sent to him and samples were analyzed in our lab on the microscope as well as our imaging flow cytometer. So I'm going to start to wrap up. So just for some final summary points on what is happening with the bigger picture of microclots and the presence of microclots. We believe that fibrinolytic microclots are widely present in all chronic inflammatory diseases studied to date, and also in healthy individuals. These diseases include Alzheimer's, Parkinson's, rheumatoid arthritis, and infections like SARS-CoV leading to acute COVID and long COVID, also ME/CFS. They can stain with various fluorogenic dyes, such as theoflavin T, or as I mentioned, amitracid dyes. They are common and in the size range of between 2 and 200, but they can also be very much larger than that. They might also be resistant to the normal process of fibrinolysis, and some are even resistant to trypsin, depending on their molecular content. They can be induced in vitro in the lab, in whole blood, in platelet-poor plasma, and also in purified fibrinogen. And we can induce them by adding a variety of substances, including bacterial lipopolysaccharides, lipoteichoic acid, even estrogen, and obviously spike protein, and as I previously showed serum amyloid A. But they can exhibit considerable structural and perhaps even spectral heterogeneity. And this structural and spectral heterogeneity will reflect the molecules that were bound to this fibrinogen or to the plasma proteins before they polymerized or changed into a insoluble little microclot. So therefore, ultimately, microclotting and endothelial damage point to the presence of a heterogeneous and very complex disease pathology in long COVID. If we compare long COVID to other inflammatory diseases, and if we look at what the differences are, perhaps that's the question to rather ask, is to say, what are the differences between the content, the activity, and the biochemical characteristics between long COVID and compare it to diabetes, cancer, and any other of the inflammatory diseases? So it's not simply a question of size and numbers of microclots present in healthy versus long COVID or any other disease with any circulating inflammatory molecules. We obviously cannot ignore the role of hyperactivated platelets and their role in inflammatory diseases in general, but their role in long COVID as well. There are wonderful research that is coming out from Professor Bonsall and from other researchers looking at platelets in long COVID, and where people even suspect that platelets might be carriers of the virus itself, and viral persistence might be involved on the pathway of platelets and incorporating the virus inside the platelets. Ultimately, the two pathologies that I just discussed here, namely platelet hyperactivation and microclot formation, might be one or two of the processes that eventually drive a widespread thrombotic endothelitis in long COVID. With that background, we have some exciting next steps planned. We are planning commercial endeavors where we are looking at imaging flow cytometry and microscopy, microclot testing at various facilities, not only collaborating researchers, but also in various clinics and various pathology labs. And our startup company, Bioco Technologies, are helping these various clinical applications to be rolled out in pathology labs and clinicians worldwide. We need to look further at the ranges of the microclots present, not only in long COVID, but also in other inflammatory diseases. We have been doing some collaborative research with the WUS lab, where we received the WUS lab samples, where they looked at previously the microclots found, or the deposits, amyloid deposits found inside the muscle samples. We are looking at their blood samples currently. We are also very lucky to have received a POTS grant. And so we're going to look, or we're busy looking at POTS in long COVID before the pandemic, so pure POTS, and then POTS induced due to long COVID. And we have got collaborators all over the world sending us samples, and we're also going to collect samples in South Africa. So one of the important collaborators here includes a collaboration with Professor Satish Raj, and he's a cardiologist from Canada and a former president of the American Autonomic Society. He's just sent us his samples. It just arrived in my lab. So we will hopefully also be able to look at their type of samples. Then we've got various other collaborators, including a possibility to look at the link samples from the group of Professor Michael Peluso and Professor Steve Dix from the US. We are embarking on more proteomics analysis. We need to find out if there perhaps are other molecules that we have missed in our previous proteomics analysis. We are comparing different other groups. We are also looking at symptoms that arrived after vaccination. That proteomic study is underway and nearly ready for our first publication to be preprinted. Then we will continue to identify more novel markers. Perhaps endothelial debris markers, perhaps biofilm markers might play a role. We need to also convert our net marker discovery, where the nets are entrapped in and structurally entwined in our microclots. We need to convert that to a method for imaging flow cytometry. And then I'm very happy to say that there are clinical trials that are planned and underway to look at anticoagulation as one of the treatment options, not the treatment options for long COVID, but in a subset of patients, anticoagulation might have some relief for some of the symptoms. And then we are busy with various collaborator grant applications. Most importantly, I think we need a significant awareness where we need to collaborate with various groups. We need to gather our strengths. We need to manage our challenges, and we need to elevate our excellence of research and collaboration, not only for research purposes, but also for the numerous patients that are in bed and debilitated without any options. Then just lastly, I'm very glad to report that it seems as if the WHO, they are really focused on guidelines and development of possible guidelines for long COVID. And I am on this panel where we will develop guidelines for the possible treatment of long COVID. Then I'm also very happy to report that I've been part of the World Health Network. We've just completed Adelphi, and we watch this space. Hopefully it will be out in public soon. And then we are busy getting people together. We've got the funding. A pediatric clinical trial will happen in the UK. We are just finalizing all the different parts of the clinical trial. So with that, thank you very much for allowing me to speak to you and to discuss all our new endeavors, to discuss some of our issues, to convince people that microclots are indeed clots, and also to convince people that we should look further, we should embark on more collaborative research, and mostly we should embark on gathering our strengths to elevate this research as a possible answer to to millions of people who are very, very ill. Thank you very much.